introduce the first presentation. Our first speaker today is Dr. Mike van Amburgh of Cornell University. I believe you all know him. He's professor of nutrition of milk cows at Cornell University. He is the founder of CNCPS. So he is the founder of a feed model that has been adopted all over the world, and he will talk about optimizing feed efficiency with amino acids to increase the production of milk cows. Well, today it is also Mike's birthday, so let's give him a big round of applause to celebrate his birthday.
Yeah. Okay. Good to see all of you. So I'm going to talk about, I don't know which one, I'm going to stand on this side. So I'm going to describe our new acids. There, that's better. And I'm going to recognize my co-authors, Andrew and Alex, who have helped a lot with some of the thinking around amino acids and protein utilization. So there's a couple things, one of the first things that we're faced with, whether we want to or not, is the fact that there's an environmental and human health aspect of excess nitrogen excretion. And cows are really central to that in some regards. If you look at it on a global basis, 80 to 81% of all the ammonia emissions come from agriculture. And we talk about methane. I don't like methane as a topic. I don't think we're going to change it much. But something that I think we can work on is reducing nitrogen excretion, because we can make diets more efficient. We can focus more on amino acids. We have a better understanding of rumen nitrogen requirements, which means we can be more precise about how we do that, which reduces urinary nitrogen excretion. The other thing I think we have to do is we have to think about the cows and how they're changing. I'm going to talk a lot about this afternoon, or in my second talk. The genetic capacity of today's Holsteins is extraordinary. I'll reinforce this a couple times, but we have first lactation cows in New York, in commercial farms, highly selected Holsteins, hitting 7% milk fat and 4% true protein. Okay? Sounds fantastic, doesn't it? Yeah, that's a whole talk, right? But what that tells me, is somebody who works on understanding the requirements of a cow, is that if they're going to produce that much fat and protein and maintain milk volume, their nutrient requirements just increased, which means that maybe the numbers I'm going to show you in a few minutes, which we derived 10 years ago, are actually outdated, right? And we'll talk about that. I think the other thing that's important here, historically in dairy cattle, when we think about protein synthesis, that's almost a linear function down to milk protein. And that's biologically incorrect, right? Everything the mammary gland does is a protein synthetic function. Lactose synthesis, fat synthesis, and protein synthesis all require enzymes, carrier proteins, transport functions. That's all amino acids. So our amino acid requirements need to be global, not specific to one function. That's one of the things in the US where our NASEM, the 2021 NASEM, failed a little bit because they drove all the amino acid requirements and efficiencies through milk protein. And it's really difficult to figure out how to reconcile that. I also would suggest that the concept of nitrogen efficiency is energy dependent, and in ruminants might be related more to urinary nitrogen excretion than how much intake nitrogen moves into milk. And then we'll talk about this, but if she's going to excrete 200, 250, 300 grams of nitrogen in the urine, she essentially told us, you overfed me or you gave it to me in the wrong form. So I don't worry about the efficiencies of amino acids to the mammary gland. I worry about what's actually being transported and cleared by the kidney, because that's the stuff that we need to get rid of. That's where our inefficiencies are at. We're not going to change the efficiency of use at the mammary gland, right? We're going to find the optimums and we're going to work around that. But we got to get rid of the stuff that she's going to excrete. There's a lower limit to that, but we've got to find a way to work at that limit. So as you can tell, this efficiency isn't just related to milk protein. It should be related to energy-corrected milk, as all those components require protein as part of the synthetic process. We just haven't really thought about it that way, and we haven't used the information in that regard. We were trying to come up with a way to describe this a couple years ago, and Alex Benoit, came up with this. I don't like talking about cows as a machine, but I think this helps us recognize kind of what happens in a cow. So here's my point. So I think meeting amino acid requirements improves overall nutrient and energy use efficiency. I think amino acids are not about nitrogen efficiency. Amino acids are all about energetic efficiency. If you think about how we're making things, how the mammary gland is synthesizing things, right? It requires amino acids, it requires some ATP, some carbon, some reducing equivalents, some glucose. For the most part, it's always on. So this conveyor belt is always running, trying to make either lactose, fat, or protein. If one of these things that it needs are missing, it's still spending the same amount of energy to run. It's just not effectively making the end product. So if we don't do a good job of meeting all of these requirements as this process is running, we lose energetic efficiency, right? So my way of thinking about this anymore is not about nitrogen efficiency. It's about how do we enhance the energetic efficiency of the cow, because she's always going to be running these things. We just have to make sure that everything's there to meet the requirements to produce whatever she wants to prioritize. So a little bit about urinary N, things like that. Quite a while ago now, Ryan Higgs spent his entire master's degree trying to figure out how to best predict urinary nitrogen and fecal nitrogen. And I show these papers just as an example. If we look at intake nitrogen, there's two studies lined up here. We go from 429 to over 750 grams. That's over a 300 gram increase. If we look at nitrogen excretion in the feces, it's fairly fixed, right? We're not going to change fecal nitrogen. If we do, it means we fed a lot of indigestible protein, right? That's also very expensive. And we probably need to know that, but that's not where the variation is, right? So we can kind of park this one and say, we don't really need to worry about this. A cow is going to excrete a fairly constant amount of fecal nitrogen. Where the variation is on the urine, right? And we can see, 93, that's pretty low. 279 is pretty high. A lot of cows operate in here. The question is how low can we go? How efficient can we make them? And we can predict this, and we've been able to predict this with fairly high accuracy for quite a few years now, right? And what you end up doing is having to iterate on the rumen nitrogen versus the MP versus the urine. So if you think about it in this regard, we published this a few years ago now. These are studies where energy was first limiting. And if you think about most dairy farms, when we go in to modify a diet, we're almost always energy first limiting, right? then it's hard, right? You're not going to go from 70 mcals of intake to 75 mcals of intake by modifying the diet and do it effectively unless you push up the starch, and then you have probably a dangerous diet, right? So we're always energy limited, and we're always working, not always, but a lot of times we're energy limited, and we're working around the balance on the nitrogen side. So in this example, most of the cows are producing about 40 kilos of milk in these examples, about 24 kilos of dry matter intake, and the crude protein is ranging from about 14 to almost 19%. And what we have here is this is milk, true protein, or total protein, converted to a nitrogen basis so we can make the comparison. There's the fecal nitrogen, here's the urinary nitrogen. So this is a good example of where, for the most part, we're operating up here, where we're excreting more fecal and urinary nitrogen than we are milk nitrogen, and that's where we're out of balance. Now we know we're not going to change fecal, so the opportunity is, can we somehow figure out how to reduce the urinary nitrogen excretion? That's going to do two things. Protein is usually more expensive than energy. And number two, excreting nitrogen costs energy, right? So we can reserve more energy for productive uses and not excreting it. It's not urea production. There's other metabolism that's going on that costs energy, right? So if we can reduce that, now all of a sudden we've dropped nitrogen intake by about 50 grams. That's about 700 grams of soybean meal equivalent, right, that we wouldn't have to feed. And as long as the rumen nitrogen balance is positive and MP is met, now we've made space in the diet for more carbohydrates. Now we can make more microbial protein, pull more nitrogen out of the rumen compartment, and make the cow more efficient. Okay? And now you've created space so you have the opportunity to do amino acid balance, right? Because you saved some money by pulling out the proteins you didn't need. Now you can spend some money on things that are much more precise at meeting her true requirements. And if we're really good, we can do it some more. And we're not always focused on urine per se. We want to talk about productive N. It's not just milk nitrogen, because we need nitrogen for maintenance, for fetal growth, for body growth, for things like that. So we talk more about productive nitrogen, not just milk. And we've had these predictions in the model for a long time. We look at productive nitrogen to total nitrogen, productive nitrogen to urinary nitrogen, and this is a very high ratio. But it doesn't really matter which model you look at, which way you look at it. We're trying to target, if we're going from 0.7, we'd like to get to a 1-to-1. You're doing great if you get to 1.25 to 1. We're actually working with some dairies right now on an experiment where we're pushing this relationship. We'll see. The cows aren't losing any milk, so far we've managed to make them quite efficient. But that should be our objective, right? I'll focus on the mammary gland now, but I think this is where we've got to start, because this is what society says they don't like. So if we move now and start thinking about amino acids, this was a comment. Gerald Lobley, who was at the Rout Research Institute, spent his life on metabolism in animals and in humans. He touched everything. He wrote this paper that I thought was really interesting, and it helped solidify our way of thinking around this interaction. So he wrote this paper called Protein Energy Interactions, Horizontal Aspects. Right? And I'm going to skip some slides here eventually that would talk more about this, just in the interest of time. But overall, what he had stated here, this quote I think is important. It's been traditional to consider protein and energy metabolism as separate entities, right? And the way we formulate diets, we've kind of had energy in one column and protein in another column, and neither did the two meet, right? And he's implying here that we recognize this as artificial, and he would say they should be considered together because that reflects how nutrients are ingested and utilized as part of normal feeding patterns. So, this basically said somehow we have to figure out how to merge energy and protein in a way that links it to the way they actually metabolize these things. And that gave us some good ideas and kind of gave us the license to think a little bit differently. The other thing that we have to consider is that amino acids, essential amino acids go in different directions, right? It's not always to a net protein outcome. And Mepham published this in 1982, and he had these group 1, group 2, and group 3 amino acids. And the group 1s, histidine, phenylalanine, methionine, tyrosine, and tryptophan, they have an efficiency of 1, which means when the mammary gland takes them up, they excrete them 1-to-1, right? So it goes in and it comes out. You have these group 2s, right? Like some of the branch chains, lysine, arginine, threonine, that go in more than they come out. You've got these group 3s that go in less than they come out, right? Well, these are mostly non-essentials, and they're being made by the group 2s, right? Plus, there's other uses for things, and that's what I'm going to talk about, right? Helene Lapierre from Canada, we would have these difficult conversations, and she would say to me, why, if lysine is a limiting amino acid, does the mammary gland take up every unit that it sees if it doesn't come out in the milk protein? And I would say it must have other biological requirements in the mammary gland that we don't fully understand. I'm going to talk about that here in about 20 minutes because all of a sudden we figured out that lysine is very important for milk fat synthesis. And we found that by accident. Right? But that's the way we learn, right? The other thing we have to think about, and this is where we're progressing in our thought process. We all, you guys are 70 to 80% of your milk goes into cheese, right? So you want as much casein as possible. If we're going to make casein, we've got to focus on the branch chain amino acids. But it's also interesting to realize that if we want more casein, we have to have proline, right? So all of a sudden, I'm introducing the idea of non-essential amino acids. The other recognition that we've made, especially with these high-producing cows that we have today, is that it's very possible that we're hitting limitations on non-essential amino acids. And if you want to make more casein, you have to have more proline. Well, that's going to come from arginine and a few other pathways. So maybe arginine needs to be considered more important than we've recognized because that may be part of our limitation on why we can't make more casein sometimes, right? And if you look at casein and you look at the branch chain amino acid requirements, it's 208 grams per kilogram. Look at proline by itself, right? So and proline gives it the three-dimensional structure. So then we're at the point where we've argued with ourselves about some of our studies where Dave Barbano would look at me and say, I think you're proline limited. And I would laugh at him and I said, I think we probably are. We made the same calculation yesterday. And that's an interesting discussion, right? So because the proline has to come from essential amino acids. And when these high-producing cows are running as fast as they can, that may become a nutrient limitation because they're using all the essentials for some other things, and converting to a non-essential may be too much of a demand. You guys see where the tension is there? So we're in the process of thinking about that. So what does that mean, right? Well, we have to get to this efficiency of amino acid use. And historically, if these are our amino acid requirements, we looked at the net requirements. We looked at milk, pregnancy, growth, reserves, and all the maintenance functions. But as we just talked, some of those amino acids could be oxidized. Some can go into anaplerotic reactions. There's a lot of those amino acids that have to be synthesized to the non-essentials. We call them non-essentials, but they're still required, right? And we've kind of forgotten that. we assume that if they're not essential, somebody's going to make them for us. And I think that's probably wrong. And then they go to gluconeogenesis. The point here is that our efficiencies of use have to be more global and incorporate all of these functions. So that's exactly the approach we took as we went back to figure out what we call the optimum requirement for amino acids. So we have this data set that we put together. We keep building it. It's similar to what Lorraine Depel published in 2004 and what Helene Lapierre talked about at the Cornell Nutrition Conference in 2007, right? So this looks dated, but it's a really powerful data set. And so what we did is we looked at the predicted requirement for methionine, right? And we estimated the requirement from the model, and we plotted it against the digested methionine, and we see a reasonable relationship here in that data set. And what we did is take the solution to the second derivative. So we're looking for the point on the curve where it's moving away from productive function as fast as possible. And that's that point right there. So that allows us to, find the digested methionine and the requirement and calculate an efficiency. And then what we did is we took the efficiency here, the efficiency of use of the methionine from that data set, and we plotted it two different ways. We plotted it as a function of its digestible methionine supply, grams of methionine per mcal of Me, or we plotted it the way that we've always done, the supply in grams of methionine per 100 grams of MP. And I think you can, there's a reasonable relationship here, but it's much more scattered. When we plot it as a function of the energy, it's much cleaner. Energy describes the requirement for methionine much more precisely than trying to describe it as a function of the MP supply. So we did the same kind of math. We found the point on the curve where we're moving away from productive function. That gives us an efficiency of about 0.57. And it says we require about 1.14 grams of methionine per mcal of Me. Very discrete, just like a pig. We did the same thing for lysine. And you can see, here's the lysine described on an energy function. Here's the lysine described as a function of the MP. And I think it becomes pretty obvious that if we're going to do this, we're going to improve this prediction, we're going to describe it as a function of the energy, right? And then we continued to do this for all 10 essential amino acids. Okay, so we just went right down the line. But again, it gave us a very discrete line, 3.03 grams of lysine per mcal with an efficiency of about, 68%, whatever that is. That leads to a table like this. Here's the R squareds on all of that information. Here's our efficiencies. The only data set that evaluated it similarly to what we did was from Helene that she published at the Cornell Nutrition Conference and talked about. So that allowed us to look at how did our efficiencies and her efficiencies line up. And for the most part, they look pretty good. There's a little bit of a difference in leucine. Our methionine is lower. She's asked me about that. I think a cow has a requirement for cysteine, but we don't talk about sulfur amino acids. We talk about methionine. So I think this is lower because it's trying to capture the cysteine requirement. Phenylalanine is about the same. Threonine a little bit lower. She didn't have tryptophan. So for the most part, we line up on almost all of these. It makes it very discrete. There's the grams per mcal. We are our own worst critics, right? We're always going to look at the literature and to find out if this looks like anything that's been published before. So if you convert it to a percent essential amino acid, the lysine's 15.1, the methionine 5.7. If we look at what Rolquin and Schwab published in the 90s, Henri was 14.7 and 5.3, 14.7 and 5.3. Okay, we're in the neighborhood. Chuck Schwab, 14.9 and 5.1, 14.9 and 5.1. Okay, says we need a little bit more methionine than they suggested, about the same amount of lysine. That gave us some comfort that even though we used very different mathematics to get these requirements, it says that we've known a lot about methionine and lysine for many years, right? We just have a different way to describe it. But it also would suggest, hey, the rest of these numbers probably aren't that far off, even though we've never had them described. Okay. Let's see, extra slides. So, but as I said, we are our own critics. That's really hard for people to believe that our modeling process is we will derive that stuff, but the next thing we're going to do is we're going to challenge those numbers, right? And the difference between what we do at Cornell and what every other nutrient requirement body does out there is that we get to touch cows, right? A NASEM, an INRA, an ARC, a feed into milk, whichever one you're using. The committee meets, they look at the data, they build a table, they make a model, and they release it. And they never test it on a cow. And then we're all left to figure out how it works, and where it doesn't work. The fun part about this thing is that we can do the same thing, but now we're going to take it out and we're going to feed some cows. If the cows agree, okay, good, we can kind of park that. If the cows don't agree, then we're going to go back and figure out where it needs to be fixed, right? So we keep iterating on the crazy thing. This is an example of that, right? So Brian Higgs, does this, when Ryan was a PhD student is when we did all of this math and started to build version 7. Andrew Lapierre follows him, and we asked the question, when you look at those data, there's a distribution around our optimums. Maybe, we say the requirement's 1.14, but maybe if we didn't get our math right, the requirement could be 0.9, 0.92, 1, or it could be 1.3 or 1.4. So what we did, to the best of our ability, for all 10 essential amino acids, is we used this data and the standard deviation to challenge our numbers. So we created a study where we used the optimum, we went down 1 standard deviation on all amino acids, and we went up 1 standard deviation on all of these to find out, what are the cows going to tell us? Are they sensitive to this formulation? Okay. So we did this like dairy farms would do this. The other thing that we are conscious of and we're sensitive to, I think sometimes when you tell them you're doing this level of amino acid balancing, that we're just dribbling these things on top of a cow to make sure they got exactly that. And we don't run our research that way. These are those cows. That's that experiment. We put it through a mixer wagon. We have a feeder just like everybody else. We have a diet. So all the noise in mixing, all the error in dry matters, everything's in this, right? This is not a really, it's not, one of those studies where everything is tightly controlled. It's controlled to the amount that we can do it, right? So this optimum diet, the control, 14.8% protein, okay, looks low to everybody. We made sure that the rumen nitrogen balance for all 3 studies was held constant and in excess, right? So rumen nitrogen should not be considered limiting here. So what you're going to see, so that says, hey, you can meet the rumen requirements, balanced for all essential amino acids, and still be under 15% protein. How's that, right? It's intimidating, right, to think that we're going to go out and do that for everybody, but that's what the cows say. You go down 1 standard deviation, we're at 14, that's our negative. We went up 1 standard deviation, that's 16.3, that's our positive. Again, isocaloric, ME excess. We did analyze several of our protein substrates for digestibility, intestinal digestibility, and we did update some of the amino acids. The one thing we did want to make sure is that we understood the amino acids before we went and did this study, so we did a little bit more work to figure that out. If you want to control nitrogen intake, which is what we were trying to do here, you feed a lot of corn silage, a little bit of high moisture corn, some triticale is another forage. You can see, in lots of conversations this week, I want to be completely, when I show these studies, I want to be completely transparent about what we did, right? I think if I publish this, I have to be completely transparent. So if I'm going to go out and talk in public, I want to be completely transparent, because even if I'm not at the talk, you can just go look it up, right? So let's just be transparent. As you can see, we've got amino acids, we got some sugar. We can feed blood meal, right, to get our lysine and methionine, forages, vitamins, minerals. There's our protein. So this is the actual numbers, 14, 14, 7, 16. So I told you what we formulated to. This is what's actually fed. So 14.0, 14.7, 16. ANDFOM, 31 to 32. Sugar around 4. Starch around 29. The starch, this is not what we advocate for starch. It's what happens when you're trying to control for nitrogen, right? Something's got to fill the space, right? So that just happens, but that's not where we would tell everybody to formulate. Crude fat, ether extract, middle threes. Rumen ammonia is the same. RDP is the same. mCals are the same. Oops, going the wrong way. Hello. There we go. So if we look at the amino acids, here's our control. If we look at lysine, we went down. That's 201. We went up 248. Methionine, 74 to 69 and 88. We found this is really hard to do, right? This is the first time. We did it once in Ryan's study, but this is really the first time that we said we're really going to try to balance around all 10 essential amino acids. And it took us about 2.5 months to figure out how to do this the first time. Involved a little bit of scotch sometimes, but we got it done. And for the most part, we got separation. Sometimes we didn't, right? Every once in a while you find it didn't go the direction you want. So what just so everybody understands how we get these numbers, we formulate the diets, we run the study, we feed the cows, we measure everything the cows do, we summarize all that data, we put it back into the model by pen, by week, and we rerun everything. And that's what I'm showing you, right? And we call that our model-guided research approach, where we may formulate it a certain way, but like everything, you don't know if you met your formulation goals. So what I'm showing you here is, even though we thought we met all the formulation goals on this side, we didn't meet the isoleucine requirements for this treatment. We probably didn't meet the tryptophan, right? Everything else seems to separate pretty well, but this helps us learn, what are the cows telling us about some of these numbers and what really happened. So when we look at milk, I'm sorry, this is all in pounds, we can see a significant response on milk yield from negative to control, from control to positive. Numerically, yes, but nothing really. Energy-corrected milk, positive response from negative to control, nothing to the positive. Fat-corrected milk, same thing. If we look at true protein yield, a big jump, nothing. A big jump, no difference. All right, so what we see here is that the optimums actually look pretty good, right? If we go down 1 standard deviation, the cows told us we're not going to perform so well. But if we go up 1 standard deviation, the cows said, I don't really care, right? Because by the amount of energy you fed me, I don't need any more amino acids. Right? So it kind of solidified, at least for this first generation model, that we can all start with those optimums and feel pretty good about meeting their requirements, right? And if we look at, this was interesting, again, this is not where we don't make a big deal about this, but we found it interesting, the milk nitrogen to feed nitrogen relationships, the best and significant difference was on the control. We had the highest efficiency on the control, right? When we went down one standard deviation, we lost efficiency. When we went up one standard deviation, we lost efficiency. The optimums gave us the best efficiency. Okay, that's good. Again, reinforcing the idea that maybe these numbers are okay. Now the problem is what do we use for branched chain amino acid ingredients? I don't know the answer either, so that's okay. But what we also do then, and this is where version 7 gives us more information, right? So we go back and we look at the control diet. It will, we look at the observed milk yield, the energy-corrected milk yield, the rumen nitrogen balance. It gives us rumen nitrogen, tells us the RDP. And because we have rumen volume, it actually gives you a rumen ammonia value. So it says our average rumen ammonia value is 8.6 milligrams per deciliter. That's great. 5.5 is your threshold. But it also gives you first limiting essential amino acid, the efficiency of use for each amino acid, and individual essential amino acid allowable milk. So in the control, our observed milk yield was 40.3. Said methionine was 41. Lysine was 40.4. Okay, so that's pretty close. But it says there's another amino acid that's most limiting. It's at 40. What is that? Well, if you look at the list and you come down the list, there's lysine at 40.4. It says valine's first limiting, right? So I'm not going to argue whether it's valine or lysine. At this point, I don't care because they're probably both first limiting and they're co-limiting. But I think this is interesting because many times I've had people tell me I amino acid balance and I didn't get a response, right? And most of the time anymore, part of that is how much did you actually feed of the amino acid? But the next point is there's probably another limitation that you can't see, right? And I think this approach is going to help us overcome that because we wouldn't have known or thought about the fact that valine was first limiting, right? We focus on methionine, lysine, and histidine, but all of a sudden we learn in this particular treatment, valine and lysine are probably co-limiting, okay? If we look at the negative diet, it's the same kind of thing. The cows produce 37 kilos. Rumen's good. MP milk says 37.1. Okay, that's troubling. Amino acid allowable. Look at this, 34. The methionine says there's valine at 36.7, so that's close. And the methionine is at 34.6, right? So that tells that looks pretty low. That tells us that we're probably at that level of methionine at the limit of what the model's capable of dealing with. And those are some things we're going to learn where the boundaries are at on the precision around predictions. But it says when you're really deficient, it may overestimate that. But again, valine's coming up. Lysine's pretty much in the same ballpark. So this says, hey, we've got some limitations here. And we knew that because that was our negative diet. So anyhow, we've got that. On the positive diet, this was, again, we learn as we go. Observed milk yield 40.7. Rumen's in really good shape. It says essential amino acid most limiting is 40.7. Okay. Ends up being isoleucine. Okay, more information. We wouldn't know that. We've never seen that before because we've never had the ability to do it. We would think, hey, we met the lysine requirement, we met the methionine requirement, we've met the histidine requirement. Why are we not seeing more milk? Because we have another what? We have another limitation that we can't see, right? So this is where I hope version 7 is going to help us all out. Because there's things that are missing and we just haven't been able to see them, and that makes us question whether The amino acids work. I did everything you told me to do on methionine, lysine, and histidine, but I still didn't get the response. Well, and we can all scratch our head and yell and scream, the amino acids don't work, the model doesn't work, the cows, whatever, there's something wrong. Maybe it's just that there's missing information. Okay. So what that makes us think about, if there's any place where the NASEM and the CNCPS agree on all of this about essential amino acids, is the prioritization. They put in the book methionine, lysine, histidine, leucine, and isoleucine. We fully agree with this and pretty much agree with this now, because as we rerun a bunch of diets through version 7, we know how to do this really well. We're finding that the next limiting amino acids are leucine and isoleucine, just like these guys identified. But as I mentioned about proline, a long time ago, Essie Evans, none of you would know her name, but she started to develop a model in the 1980s through the 90s for Nutrico in Canada. And Essie, she never told me a lot. She didn't want to give up her secrets, but she would always tell me arginine's your most important amino acid. And I studied arginine a lot as a graduate student, and I probably couldn't quite figure out why she would say it was more important than methionine. But now that I understand milk synthesis and some other things around immune system, blood flow and regulation, things like that, I would have arginine somewhere on this list, right? I'm not worried about valine. I showed you that valine limitation, and we think about branch chains. Very rarely do we see that one coming up, but the leucine and isoleucine do show itself. Okay. Also, as we were building version 7, a few people said to us, it's going to take you a while to get this out. Can you take those derivations for amino acids and run them through the current version that everybody uses, like 6.5, 6.55, 6.56? And we said yes, right? And the three that we had the most faith in were the methionine, histidine, and lysine. And when we re-derive those, each model has their own specific targets, right? You can't use version 7 in anything else because it solves its amino acids differently than everything else. So methionine, 1.19 grams, histidine, the same, lysine at 3.2. We have feedback now from very good nutritionists in the U.S. dealing with modern high-producing Holsteins that would say, your histidine should probably be a little higher than the methionine. That's really hard to do, but they're telling me that, if you're going to feed 80 or 85 grams of metabolizable methionine, you need to have the histidine about 10 grams higher than that. So this should probably be reconsidered, and I think that's the fun part about this effort, is that we can go back and figure out how do we make this more precise as we go. So I'll talk about this study later, but this is an example of how this all works. So if the cows are consuming about 71 to 72 mCals, it's straight math. That means they require 85 grams of metabolizable methionine, at least 85 grams of histidine, and about 229 grams of lysine. That lysine number scares everybody, right? Because it's much, much higher than anything we've talked about before. But every time we get close to those numbers, the cows will tell us something, right? They'll make more protein, they'll make more fat, they'll make more volume, but those numbers seem to be working, and I'll demonstrate that. It's my water. What's time? No, when am I supposed to stop? Just keep going. Okay. Yeah, I know. So the point in this slide, there's a couple things in this slide. And the reason I'm showing you some cow data is what I want you to know is that we've taken those amino acid numbers that I just showed you. We've taught a lot of our nutritionists in the Northeast U.S. and in the upper Midwest how to use them. And when they apply them and they make a few other adjustments to the diet, the cows respond, right? So this is a 4,000 cow herd in northern New York. They've got 3 bulk tanks. In this test, they had Holsteins and Jerseys. They just sold the Jerseys. They don't have them anymore because the Holsteins are way beyond them. This is the April test, right? The week of Easter, actually. They never dropped below 4.4. Actually, I have them at 4.2 here. They never dropped below 4.4 through the summer. And they're back up to 4.8, 4.9 now. So in April, the overall farm butter fat was 4.9. This is true protein, 3.5. So crude protein would be 3.7 to 3.8. Okay, just so you guys know, they're averaging about 44 to 45 kilos of milk. So they're making good milk. Here's bulk tank number 1, 4.89. Bulk tank number 2, 4.87. Here's the Jerseys, 5.43. Here's the Holsteins on protein, 3.53, 3.54. Jerseys are 3.66, right? So what we're seeing is that the Holsteins, when fed right, are starting to do what? Challenge the Jerseys for components and have about 20 to 30 kilos more milk, which is why he sold the Jerseys, right? But he also happens to have a barn. So this is what I was referring to. He has a barn of genomic Holstein heifers where he does superovulation, in vitro fertilization. He's really shortened the generation interval, and we've got them on a pretty good diet. At least 40 kilos of milk. I have over 5.2 fat. He sent me some data just before we left. I didn't update the slides. This group is averaging 5.6 fat right now. 5.6. They're hitting the high cows and the high heifers in there are hitting 7 on fat. So I've got to update all these numbers, right? This is kind of crazy, right? That's a whole nother discussion, right? We don't have the time to get into that. The reason I'm showing you this, though, is that when we use these numbers, the cows are telling us what? We can do really good things, right? It's not, and this is going to sound funny, and I'm not going to talk about this. In mid-lactation, what we see, in established lactation, what we see cows doing is increasing their components. If you want to change milk volume, right, and I can figure out how to pull these slides in later. If you want to increase milk volume, you have to start prior to calving with amino acid balancing, and you have to do it during the first 2 months of the lactation. And what we know we can do, there's some studies from Ohio State and a study that we ran at Cornell that we're going to rerun. You're talking about 7 to 8 kilos more milk, right? So you can drive volume if you start at the initiation of lactation. If you're already in established lactation, you increase your components, which increases your feed efficiency because they don't eat more, right? So your margin still goes up, but if you don't get paid for the components and you get paid on a volume basis, the only way to do that is to start early, okay? So we can come back to that during the discussion. Here's another herd. This is an incredibly well-managed herd in my neighborhood. I'll come back and talk about this herd later. They're making just over 15,000 on average milk. Bulk tank's been running 4.9 to 5, and true protein 3.5 to 3.6. There are high cow groups, over 72 kilos of energy-corrected milk, and the feed efficiency is 2.1. And I think that feed efficiency is really important. One, you notice we're doing it on an energy-corrected milk basis, right? Milk volume is not capturing all the information, right? Because if we can get these cows that are making that much milk, but making those components, and they don't consume any more feed, that's the interesting thing, as we feed these cows more amino acids and the right amount of amino acids and not too many fatty acids, we see their milk components increasing. Dry matter intake is holding, right? So you just see this increase in feed efficiency. They don't have fat cows. They don't have any fat cows because where'd they put all the nutrients? in the mammary gland, right? So when I see fat cows now, what that tells me is at some point in their lactation, we didn't give them the nutrients that allowed them to partition to the mammary gland. So they're going to be like all mammals, and they're just going to put it on their back. But if you got fat cows anywhere, all that's telling you is you missed the opportunity to allow the gland to fully express itself, and she's just going to keep those calories for the next time. But I also think this begs a conversation, because as I go around the world, I'll hear dairy producers say, well, I tried amino acids and they don't work, right? And I'll say, what did you do? And they'll say, well, I fed 10 grams. I said, 10 grams of product or 10 grams of metabolizable amino acid? And it's generally 10 grams of product. And I'll say, you didn't do anything. Well, I fed it and nothing happened because you didn't come anywhere close to meeting their requirement, right? So if I take the previous herd and I assume 59 kilograms of energy-corrected milk, which would be not a bad number for a portion of the herd, that's about 86 mCals of ME. That means 102 grams of methionine and histidine and 275 grams of lysine. Right? And I show this just to have us think about what do these numbers really look like in high-producing cows that are eating well and have high energy demand. They're much higher numbers than we've ever considered. But this is their metabolic demand. Right? So I think, when somebody says, well, I'm just going to feed 10 grams of some product and see what happens, save your money. Right? That's not going to get you anything. Right? And you can't just feed one. It's the other thing we got to get at. You could feed all this methionine and not do that, but what's your response going to be? Only as good as the limitations here. So you kind of wasted your money on the methionine, right? That you can't get magic from one amino acid, right? They've all got to move together. It is a requirement for protein synthesis, okay? I think that's the other thing we've got to get our head around. So amino acids are not just for milk protein. I have 5 minutes. Okay, well, we'll see where this goes. I'm trying to go slow because I have a translator, and he was very adamant that I not talk fast, right? This is, it's actually taken a lot of work to breathe differently so I don't talk fast. So the efficiencies and requirements can't be linked to milk protein. We have to think about amino acids in many different ways. All right, I'm going to skip a few slides here. So real quick, where did this, when we see these milk fats, what does that mean, right? Amino acids, supplementing amino acids, and all of a sudden we get more what? We have more milk fat, right? Is that intuitive to us the way we were all trained? No. What if I told you I can add a certain profile of fatty acids and get milk protein? It's just the opposite, right? I can explain all those mechanisms now that we understand it. Here's one of the things that I want to help you understand that, right? And I'll show you the study this afternoon, but we are constantly learning, right? These cows keep challenging what we think we know, and we got to go read the literature and do research to figure it out. So this is the effect of lysine in bovine mammary cells on fat synthesis, and in this particular two graphs, it's in the absence of any supplemental fatty acids. So these guys went out, they took biopsies of lactating cows, brought the cells back, and incubated them in different mediums. They looked at SRBP1 protein synthesis, right? This is one of the big regulators of fat synthesis, and you can see that as they added lysine, they saw an increase in the expression of SRBP1. And as they added lysine, they saw an expression in lipid density droplets. In other words, it was making more fat. Then they added fatty acids, and they happened to add 50 micromoles of palmitic and 50 micromoles of oleic, and they did the same thing on the lysine. And you see these nice increases just like we did before, but they're greater increases. Secretion of triglyceride, greater increase, lipid droplet density goes way up, And notice here it doesn't drop off. So when the cells have the fatty acids and the lysine, they keep going. In other words, the reason that this is dropping off is they're probably running into some other limitation. It's not that the lysine hits a maximum, it's that they don't have any other things to help do the work. Okay? When you put it all together, here's secretion of triglycerides. So this is no supplementation, lysine, fatty acids, the combination. Now all of a sudden we see this integration of the amino acids and the fatty acids. Well, this is really interesting because this is saying the gland would prefer to see a certain ratio of the fatty acids when it sees the amino acids. Well, this implies the cows have a fatty acid requirement that we don't fully understand because we've just thought about fat as a bucket of energy, and the cow's saying, no, I'd like a certain fatty acid profile, just like I want an amino acid profile. So now we've got to get more refined in how we feed fat. You do the same thing on SRABP1. Yeah, you see a little response to the lysine and fatty acids. You add them together, boom. Fatty acid binding protein really doesn't care. It just says, hey, just give me something. Okay. So fatty acid synthetase, if we look at, which is synthesizes de novo fat by elongating the fatty acid carbon chain. There's active sites all along there for histidine, lysine, serine, and cysteine. They've looked that, fatty acid synthetase is highly conserved in every tissue. They looked in human liver cells, and in human liver cells that were deficient in histidine and lysine, they couldn't get expression. When they added the histidine and lysine, the enzyme increases expression. Well, that's interesting. If you want de novo fat, it says you need histidine and lysine to induce the enzyme for de novos. And that's what two of the papers actually say. If you want fat synthesis of de novo fats or export out of the liver, you need histidine and lysine. All right, that's a linkage we hadn't seen until we went and had to look at it, right? And there's some non-essential linkages there too. So, hey, I'm going to make it. So if we isolate the nutrient requirements for nitrogen, it allows for better partitioning of what we call RDP and RUP and amino acids. The model is on a nitrogen basis. So the whole flow of the model is on a nitrogen basis, which makes predicting the amino acid supply more precise because each amino acid has its own nitrogen content. It's not 6.25. When we describe it on a gram per mcal basis, it increases the precision, and the cows like that. And then that allows for an evaluation of optimum versus maximum, right? We could formulate more amino acids for some maximum response, but likely it's not going to be financially rewarding, so we want to stick closer to these optimums. You guys will have these slides. The last couple slides are just diet guidelines that fit what we've been doing, right? And we can come back to this during the Q&A. Okay.